It is with great pleasure. I'm so excited to introduce Dr. Julie Allen. She is an assistant professor in the biology department at the University of Nevada, Reno, where she has been faculty since 2018. She received her PhD in 2010 from the University of Florida in evolutionary biology. She did postdoctoral work at both the University of Illinois in genomics and the Florida Museum of Natural History in bioinformatics. Her work integrates bioinformatics tool development with genomics, museum science, and public participation in research, formerly known as citizen science, to answer questions about biological patterns. She is advising master's and PhD students and postdocs in projects that include bioinformatics, museum science, genomics, morphology, and evolutionary biology. She is the project lead of Notes from Nature, an online public participation and research project based out of Zooniverse that has thus far transcribed over 2 million museum specimens from over 10,000 volunteers. Dr. Allen has recently become the curator of genomic resources and bioinformatics at the University of Nevada, Reno Natural History Museum, where she is working to promote outreach and teaching both within the university and beyond. Now, I've had the great fortune to know Dr. Julie Allen for quite some time, and she has been so generous with her time in speaking with Vanderbilt students as part of a macroevolutionary course over numerous years over the past decade. And so it is with great pleasure and completely fitting that we are here having her today speak to and give an evolutionary studies seminar. So thank you very much. And without further ado, Dr. Julie Allen.
Great. Thank you so much. Thank you for that kind introduction, Larissa. It's great to see you. And thank you everyone for coming and for the kind invitation to be here. Okay, I'm just checking. Can you hear me okay?
Yes, we can hear you perfectly.
Okay, great. So what I kind of would like to do here is give a little bit of background about me and sort of how my journey into getting where I got to today, because that kind of helps to explain my research program and also this talk a little bit. So I'm a classically trained evolutionary biologist. I've been really interested in co-evolving organisms for most of my PhD. And as I was going through that process, it became clear to me that to answer some of the questions that I wanted to answer, I needed larger data sets. And to work with those data sets, I needed tools. Unfortunately, a lot of the tools that were available at the time either didn't do exactly what I wanted them to do or were very kind of black boxy, and it was difficult to tell what was kind of happening behind the scenes. So as a postdoc, I moved into tool development. And a big part of my research program now is to help build software, pipeline scripts, et cetera, to enable not only researchers, but also the general public to help us collect, analyze, and work with big data. So this is kind of the format of the talk today. And what I'd like to do is kind of talk about some of the questions I answer in evolutionary biology and how I incorporate bioinformatics and tool development into that process, and then come back and talk a little bit about some of the public engagement work that we're doing and how this also enables us to kind of pull together large data sets to answer big picture questions. So the research in my lab is really interested in the focus of the interaction between ecology and evolution. So in fact, I'm particularly interested in how ecological processes affect and create the evolutionary patterns that we see. So for example, we have this little bird here. This bird is of course affected by abiotic factors. These are just temperature and other climate layers from Florida. So they're affected by abiotic factors that hit them, as well as biotic factors. So the habitat that they reside in, the predators, and of course, importantly, for a lot of what I think about, the parasites as well. So my lab, I'm really interested in how both abiotic and biotic factors come into play to help us understand large scale patterns. So for example, we can look at large spatial patterns. So this is a large map of bird diversity. And of course, there are some really neat patterns that you can see right off the bat. There are certain areas of the world where we have a lot more species of birds than others. So those are some of the spatial patterns that we see, but we also have large-scale evolutionary patterns. So if we build a big evolutionary tree of birds, for example, we can see areas of rapid radiations and other kinds of factors here. And both of these patterns are there due to the impacts of abiotic and biotic factors that are happening on the species at ecological timeframes. In this talk today, I'm mostly going to be focusing on biotic relationships and how those biotic relationships help us understand evolutionary patterns. So this, of course, is the adorable little organism that I study. This is a parasitic louse. This is a Pediculus schaefi. This is a louse that lives on chimpanzees. This louse is almost identical to that of the human head louse. There are very few people in the world that can tell them apart. These lice are obligate permanent ectoparasites. Obligate meaning they require their host for survival. Permanent that they live their entire life cycle on their host. So lice are actually really interesting because they are in two different obligate permanent relationships. They are ectoparasites of both mammals and birds, and they all harbor obligate bacterial mutualists that reside inside specialized cells. So this right here is a glowing fluorescent cell showing the DNA of the bacteria fluorescing from the abdomen. In fact, you can even see this little opaque cell shown here. This is the mycetome where the bacteria live. The bacteria are thought to supplement the louse's diet with missing nutrients and enabling them to feed on relatively nutrient-poor diets from the host, either blood or feathers. So the system for me is particularly interesting because we can ask evolutionary questions. So for example, we can ask, how does the population structure of the host affect the population structure movement of the lice and therefore the long-term evolutionary patterns? How does defensive mechanisms of these birds, for example, affect the behavior and the morphology of these lice over the long term? From the bacteria side, we can ask questions about what are the supplements that these bacteria are providing to their hosts. What bacteria become these endosymbionts? Are they in the same place? Do they move around in different species of birds? And how does that co-evolutionary system work over time? But the system is also interesting from a bioinformatic perspective. So each of these data sets has a different ecological, morphological, and importantly, for what I think about a lot, a genetic data set, each with their own challenges, shall we say? And we approach each of these data sets from different perspectives as well. So we can kind of come into the system from a whole bunch of different angles to start trying to tease apart some of these large-scale evolutionary patterns. So today I'm mostly going to be focusing on the coevolutionary history of birds and their ectoparasitic lice. These lice are thought to feed on feathers. So if you look at this feather here in the upper left-hand corner of your screen, this is a healthy downy feather, and this is that downy feather after it has been exposed to these ectoparasitic lice. So you can see these louse have actually reduced quite a bit of feather mass here, and you can imagine with a really high louse load, you can imagine quite a bit of damage being done to those feathers, which could affect the bird's ability to thermoregulate, et cetera, et cetera. So birds have developed these really cool behaviors and even morphologies in some case for getting rid of their lice. So if you see these ducks here, you see what they're doing. They're taking their bills and they're going into the wings there. And what they're doing is actually like scraping lice off their feathers, right? And trying to get rid of them. They're trying to keep their overall louse loads down. And in fact, we know that birds with really high louse loads have lower survivorship rates. So this is actually a really critical process. And if you're watching a bird sitting on a wire at any point, often you'll see them kind of do this thing where they take their bill and go into their feathers. And what they're really doing is just budgeting their time between finding food and other important biological processes, but also keeping their ectoparasite loads down. So this grooming behavior has actually been really critical in terms of what it actually does to the lice too. So this is a healthy louse shown here, and these are 3 lice after they have been groomed off of the bird. So remember, these are permanent ectoparasites. They require the host to survive. So not only do they need to not be removed from the host, if they are removed, they need to get onto another host really quickly. So this grooming process actually not only removes the lice, but also kind of destroys them in the process. So you can see this one's entire abdomen has been crushed. This one's missing part of its abdomen, and this one has completely lost its head. So this grooming process not only removes them, but also often kills the lice in the process. Because of this, lice have evolved some really interesting adaptations for trying to avoid being groomed off by their host. So this is a wing louse here. Wing lice tend to be really long and skinny, and they fit precisely between the barbs of these wing feathers. So these are a whole bunch of wing lice. You can see them fitting really snugly in between these barbs here. And this is to really prevent that bird from being able to take their bill and scrape those lice off. They're finely hidden here. So this is one adaptation that the lice have evolved into to avoid getting groomed off from their host, but there are others. So there's another ecomorph, as we're calling them. This is a head louse ecomorph. This louse lives on top of the bird here. And you can imagine that the bird can't use its bill, right, to sort of scrape the lice off the top of its head. Rather, instead, it uses its claws, right, to scratch off the top of the head or get some hapless human to kind of help them in the process. Head lice live on top of the head. They have themselves a very large head structure. And in fact, it's thought what they do is they take they have giant muscles and they clamp down on the feathers and just sort of hold on really tight to prevent from being scratched off by the bird. So this is another adaptation that these lice have evolved into to avoid host defense. A third ecomorph that I'll kind of briefly mention is this body louse ecomorph. This is kind of a plump little round louse. These lice are really good at hiding around the host body and absconding and hiding between the downy feathers to prevent getting both groomed and scratched off from the host. So these different ecomorphs are quite interesting. And when you look across different families of birds, what we find is a pattern of many families have more than one ecomorph. Right, parrots showing 3 ecomorphs here. Tinamous in particular, quite interesting. There are a number of ecomorphs that have been identified from tinamous. I'm just showing the most common ones here. So this begs the question as to how did this pattern evolve? And there are a couple of different ways that we can think about this. So one hypothesis would be that you have some ancestral bird here. It has 2 different ecomorphs of the lice. That bird goes through some speciation event, bringing their lice right along with them. Their lice then go through their own speciation event. If this were the case, then we have a very specific idea of what the evolutionary tree would look like if we built the tree of the lice. We would expect that then all wing lice here would group together and all head lice would group together. So these ecomorphs would form these monophyletic clades. A different and perhaps more interesting hypothesis is that of this one, where we have a bird. It has some ancestral ecomorph. That louse population moves into different parts of the bird's body and over generational time evolves into these different ecomorphs in response to selective pressure from the host. If this were the case, then we would expect a different pattern that we would see when we build the tree of the lice. We would expect instead that head lice and wing lice, for example, are sister taxa, but we certainly wouldn't expect all head lice to group together and wing lice, but rather lice from the same host, but different ecomorphs will be closely related. This has been a big question in the louse community for quite some time, and some of the initial trees using a few loci started kind of showing this really kind of interesting pattern of potentially looking at different radiations of these ecomorphs on birds. However, these initial trees lacked a lot of topological support, so they're using just a few markers. And the big question then is, all right, is this tree real? What is the real pattern? And of course, this is a bioinformatic question. So how many loci, in fact, are needed to build robust phylogenomic trees of lice? We know that lice radiated with birds. So in fact, when you build an dated evolutionary tree of the lice, many of these lineages go way back beyond the KT boundary when we thought that the majority of dinosaurs went extinct, leading to this idea that lice were around a lot longer than that, and therefore it's likely that feathered dinosaurs themselves had lice. We know that birds then, these that evolved into birds went through a massive rapid radiation right around this time, likely bringing their lice right along with them. And when we build these trees of the lice, we see a rapid radiation of their lice as well. So once we get into large trees of rapid radiations, we get into a really difficult phylogenetic and computational challenge. So in fact, building the evolutionary tree of birds has been one of the largest computational challenges over the last decade or so. So there have been a number of data sets that have been built using genomic data, trying to understand and trying to resolve some of these branches of these rapid radiation in birds. So this has been a problem that has been approached with many, many different types of genomic data trying to resolve this. And I'll just say that if you look and you actually squint really, really closely, you see these little tiny like white bits here. So those are actually louse eggs, and this might be the only time that lice actually make it onto the cover of Science. So this is actually pretty exciting for the louse community. But of course, this work has continued in trying to understand and resolve what the phylogenetic tree of birds are. So it stands to reason that if it is as difficult to build these evolutionary trees of birds, we're likely going to run into some of these same problems with the lice. And therefore, we're going to need genomic data from lice. So I'll just say in a very cheesy way that we're lucky in lice in that they have incredibly small genomes. So their genomes are about 100 to 400 megabases. And so because of that, we can sequence their genomes for the low price, in fact, of $218, giving us about 60 to 70x coverage. And what's really neat about this is we're not using any kind of genome reduction methods where you try to like reduce the amount of genome that you sequence so you can reduce the total cost of sequencing, but instead, we can get the entire genome for $218. And generally what we get, which is really neat with this approach, is that we get many sources of data. So we have the mitochondrial DNA, the nuclear DNA, and of course, interestingly, a lot of their internal endosymbiont DNA as well. And we demonstrated that we can in fact assemble full genomes of the internal endosymbionts from our sequencing methods, which is really neat. So the next question is, how do we assemble genes from short read sequences of these louse genomes? All right, so when people assemble genes from a genomic data set, they generally take two different approaches. So there's a de novo approach. So you take all these, when you assemble a genome, what you do is kind of chop it up into a whole bunch of tiny little bits called reads. And then in a de novo approach, what you would do is just try to find matching overlapping sequence and try to align all these little reads to each other and fill out the whole genome. Another method is called a reference-based genome, where you have a fully annotated genome and you just try to see where these reads match onto that genome. Both of these methods are incredibly time-consuming. They're computational barriers for students and for anyone else trying to get into this process. And they each have their own computational issues. For example, you can't do a reference-based assembly on anything that's more than 2 million years divergent, which is an impossibility when we're dealing with lice. Most things are well beyond that divergent level. So what we wanted to know when I started kind of thinking about this problem and getting into it is, well, can we just skip this whole process? Can we take all of these short little reads and just say, I want this gene or this locus? Can we just target the reads that are associated with that locus and assemble that locus, reducing all of this time-consuming and computationally difficult steps? So this was the idea behind the software that I developed. It's called ATRAM, Automated Target Restricted Assembly Method, and we initially released it in 2015, which is crazy for me to think about now, like how long we've been developing and working with this software. What it does is target and assemble any locus of interest, and it does it through massively parallelizing the process. I'll kind of get into that just a little bit, but not a lot. And it works in two steps. So let me just briefly go over the two steps of how ATRAM works. So the first step is to format the database. So we have all of these short reads from the genome. These then, generally what we get when we do sequencing is we have a tiny little piece of sequence from this end of a read and a tiny little piece of sequence from that end of a read. These are called mate pairs, all right? And generally what you do is you get a file that has all the first mates, and then you have another file that has their mate pairs. So ultimately what you have is a part-end sequencing. What ATRAM does is take all of these little reads, breaks them up into little clusters called shards, and turns those shards into BLAST formatted databases. What it also does then is index those mate pairs, because we know if we get a hit on one of these reads, we're also going to want to pull up its mate pair because we know they are on the same piece of contiguous DNA and they're going to be very close. So the idea behind breaking up this library into a whole bunch of different shards is for parallelization. And the idea is that you can, if you have access to a cluster with many cores, you can then say, do the next step by blasting each of these with a different core, rapidly speeding up the process rather than blasting them one at a time, for example. So the idea here was really to kind of think about how can we do this and take advantage of some of our computational resources to really speed up the process. So again, if you have a library with 100 cores or 100 shards and you have 100 cores available to you, you can blast them all at once and quickly assemble the gene. So that's the idea behind how the database is formatted. In the next step, we assemble the genes. So what we do here is we take a target sequence. This can be a closely or very distantly related a contiguous piece of DNA, a protein or DNA, that is then blasted against the entire library. The reads that match are pulled. Their mate pairs are pulled as well. And then those are assembled in a de novo fashion. And in the next step, so then we have what we call our contigs, these little contiguous pieces of DNA that have been assembled. In the next step, these contiguous pieces of DNA become the target. So in the next iteration, you take the pieces of DNA that assembled, those get blasted against the library as well. The reads that match are pulled, their mate pairs, and again, it's assembled in a de novo fashion. So this is done for multiple iterations until at the end, what we often find is that we have the entire locus of interest, but often a lot of flanking region on the side as well. So this step is done for as many genes or loci as you have to assemble. So if you have 1,000 genes to assemble, you do this step 1,000 times, step 2. So ATRAM is fast. It can assemble a locus in just a few seconds to a few minutes. I've seen a UCE loci be assembled in about 8 seconds, for example. It was written to be scalable, so it can be run on a personal computer or on a high performance cluster. And the idea is the way that we designed it is to be able to take advantage of the abilities of really scaling up on a high performance cluster to speed up a lot of this assembly process. And as I said before, each gene is an independent process. It can run thousands of, if you can run thousands of processors at once, for example, if you wanted to assemble 1,000 genes, you could hit them all at once and then assemble all of them immediately. The fastest I've really gotten it to go, only because I didn't want to ask for more resources, is about 1,000 genes per hour. But again, this is just dependent on the amount of resources that you have available to you and you can ask for. We completely retooled ATRAM and rebuilt it in 2018, and we've been kind of working on it and adding little features and whatnot ever since. So getting back to our original question of what are the patterns, these phylogenetic patterns that we see with these lice, to kind of get at this, we sequenced 38 genomes from bird lice, we assembled 1,000 genes using ATRAM, and we built the phylogenomic tree. So when we got this pattern, so if you guys can see these 100s, these are bootstrap values. We got incredibly high support across, well, the highest support across all of the trees, which was really exciting, and suggesting that like our method and our loci that we're using can help us resolve these difficult trees. And when we put the ecomorphs on here, this is the pattern that we get. So if you remember, we were trying to see, do all head lice group together into a monophyletic clade? Do all our wing lice grouped together in a monophyletic clade, right? But instead what we see is multiple adaptive radiation, so multiple evolutions into these different ecomorphs, which is really suggestive of this kind of a pattern where you have a bird, it has an ancestral louse, that louse population moves into different parts of the bird's body and over generational time evolves into these different ecomorphs. This is really exciting. There have been a few systems that have become famous for looking at multiple kind of adaptive radiations. And the most famous, of course, is that made famous by Jonathan Losos and the Anolis lizards in the Caribbean. In this system, what you have is competition between different individuals of Anolis lizards that have pushed them into partitioning their habitat and evolving into these different morphs that specialize on different pieces of the habitat. So here the selective pressure is competition. In this system, what we have is kind of a similar pattern over and over, but with a completely different selective pressure, which is host defense, which kind of really leads to a lot of fun questions now as to kind of getting into this pattern and what we can learn about it. So we received an NSF grant about a year ago now to start really diving in and resolving some of these issues with these lice. And the goal of this proposal is really to kind of, how do we explain this pattern? What can we determine from the selective pressure? What can we determine from molecular changes? And what can we learn from morphological changes that have happened here? So what I'd like to do in the next few minutes here is just kind of dive into just really briefly how we're going about answering questions about selective pressures and morphological changes. And then I would like to show you some preliminary data that we're getting with the morphological work that we're doing. So let's just start with the approach of the selective pressures. So there's a couple of really interesting questions that we can ask. So for example, how many radiations have occurred in general? So right now we're sequencing the genomes of 280 species of lice to build this tree to start really asking like how many times do we see these different radiations occur? Do these radiations occur after host switching events? And what's neat here is we can really take advantage of all the work that have been done to build bird trees to start to ask questions about, all right, how often do we see a node where we have two different ecomorphs on the same host species, right? So when we see this, do we see when we compare this tree to the bird host that kind of corresponds to a host switching event, for example? All right. So, and on top of that, we can also ask interesting patterns of how the evolution has occurred. What is the pattern of evolution? For example, do we always see, head lice going to wing lice? Or are there very, very specific ways that this process occurs? Do wing lice always stay wing lice? Is that a dead end, for example? Unlikely with the level of changes that we're seeing, but at least it kind of leads to this sort of like interesting questions of how this repeated evolutionary process can happen. All right, we're also looking at what are the molecular changes that have occurred. So very recently, in fact, in just January of this year, we got our second annotated genome in the louse world. The pigeon louse was, Columbicola columbae was annotated and published in January, and we now know that there are about 13,000 protein coding genes across pigeon lice. This is in fact one of our ecomorphs and is much more closely related to all the lice that we're dealing with. So what we're doing now, and this is work that's being led by my graduate student, Avery Grant, is assembling 13,000 genes from each independently evolved ecomorph. And what Avery's really been doing is asking questions of like, how can we speed up these assemblies, right? Even if you have one node and you can like kind of like chug away assembling these independent genes, it still does take a long time. And particularly when we start getting up to questions of, okay, how about 13,000 genes? Each one of those as an independent process, this starts to get slow, and it's slow particularly for someone who's on the PhD timeframe, and this is just a single chapter. So this starts getting, so what Avery's really been looking into is how can we speed up these runs? And here's what we're kind of thinking about now. So say you have one compute node. The original idea is that you would then use all of the cores in that node. So you have 32 cores to blast all of the libraries in the database. Then the process goes back and uses a single core to grab the top hits, the mate pairs, and do the de novo assembly. It then expands back out to using 32 cores to do all of those blasts, comes back in, uses it one or maybe just a few cores to grab the top hits and mate pairs. And this goes through iterations. And when we're kind of looking at this process, what we're finding is that on average, a lot of these cores are actually sitting idle for a lot of the time when it's going through this process. And in fact, on average, you're only really using 1 to 2 cores across this entire run. So that's a lot of wasted computational time and space. And in particular, when we're thinking about renter nodes, which is becoming really, really common in the high performance commuting world to rent time and space on a cluster, this is really actually not feasible, right? To have a lot of the process sitting idle. So what Avery's been doing is really working on methods to speed up the gene assembly. And what she found is that IO, input-output processes, really are the majority of what is happening here. So she's designed a different way to run this. And instead, what we do is say, all right, we have one core, right? And we have 1 node, and we have 32 cores. Well, let's set up 32 runs, right? Let's split up our 13,000 genes into 32 different runs and just have 1 core per gene, right? And what we're actually finding is this process is reducing days and even weeks in some cases off of the computational time, depending on the size of the library. So this is really exciting. And what Avery's doing for her first chapter is going to write a little sort of like how to efficiently assemble lots and lots of genes over a short period of time with expensive computer resources. So saving a lot of money. So that's kind of her process here. Once she has all of those genes assembled, then our next questions are, what are the patterns of selection across protein coding genes that cluster with each ecomorph? For example, do all of the wing lice have very specific SNPs, for example, that head lice don't? The next question she's also going to ask is, what is selection on branches? Do we see areas where we see like kind of rapid evolution kind of leading up to these branches? And of course, leading back to the last question of how does that relate with host switching events or other kind of large scale ecological patterns that we see over time. Okay. So what I'd like to do now is just kind of get a little bit into some preliminary data that we're finding with the morphological data. This is work that's being led by my postdoc, Stanislav Kolinchik, who's an excellent morphologist. What we often do with lice is we take them, we clear them and squish them and put them on a slide here. So you often get these kind of flattened images of lice, and which is great, right? Because you can see a lot of the different characters. For example, these CD here, you can get genitalia characteristics, etc. However, for a lot of the questions that we have here, we actually kind of really want to know more about some of these morphological features. And in fact, what we really want to know are kind of what are the 3D structures? What do those look like? So as part of this process, what we're doing is MicroCT scanning lice. And what's neat about this process is you get the 3D louse here, so you can see all of these external features as well, but you can also get internal features, right? And for example, what you can do is take all of the features. So say, for example, let's just highlight all of the muscles inside the head. The way the CT scanner works is it sort of indicates the density of the types of tissue you're looking at. So you can sort of say, all right, this is a muscle, give me all the tissues of this same density, right? And you can then highlight all of the muscles and then even isolate those, right? And ask questions about volume. And that can give you ideas about muscle strength, for example. So remember that I was kind of talking about the idea behind what the head louse does, right? So what are these specific characteristic features of each ecomorph? The head louse is thought to have, it has this huge head, and therefore the idea has always been that it has these giant chewing muscles, right? And what it does there is take those chewing muscles and then it bites down on the feathers to avoid getting scratched off by the bird. So this is the first feature that we've been trying to go after with these CT data and ask this question: do head lice in fact have larger chewing muscles as a way to avoid host defense? So what we've done is isolate all of the chewing muscles. These are the adductor muscles shown here in the head louse across a whole bunch of independently evolved ecomorphs, right? So you can see the chewing muscles here from the head louse going all the way down into the wing lice here. And the wing lice, you can see just at looking at the muscles, they're actually a lot thinner, kind of a lot more spindly, shown here. So when we look across a whole bunch of independent evolutions of these ecomorphs, what we find is this. So this is the volume of the chewing muscles shown here on the y-axis, and these are the different ecomorphs. And in fact, we see a pattern of head lice having the largest chewing louse muscles, body lice coming in kind of next with generalists, sort of like almost no real difference, although generalists seem to have a lot less variation. And then wing lice have much lower volume of chewing muscles. So this is really like exciting, and also kind of puzzled me at first as to why these guys were here and not kind of just head lice just have giant chewing muscles versus everybody else. But in fact, we kind of see this pattern of decreasing volumes. And, you know, kind of thinking about it, it sort of became really obvious to me that like you have body lice, which are thought to be prone to both preening and scratching of the bird. So it makes sense that they might need both, like they might need to use those muscles to kind of bite down and prevent. Generalist lice are also found all over the bird's body, and they're actually really good at like running around really fast. This is kind of an ecomorph that not a lot is known about, to be honest. But they also are likely dealing with the bird kind of scratching itself as well. So they also have large muscles, but they certainly haven't necessarily evolved into specializing into this one niche found on the head, where that is the only defense system they have against the bird. And same with the wing lice as well. These guys only hide by their host by hiding between those barbs of the wing feathers. So they have definitely want to be skinny and thin, and having large chewing muscles wouldn't be advantageous in that step. So it's really neat as we're kind of sequencing more and more. This is like brand new, fresh data, is that as we're getting more and more, we're able to kind of like look at males and females, for example. So how do how do those differ? And what we're finding that we didn't expect is some sexual dimorphism here. And in fact, what we find is that just, in general, males seem to have a little bit larger chewing muscles than females, although there's definitely a lot of overlap there. So that's kind of a neat new pattern that we didn't expect to see in the process. So there are a number of other kind of characters that we're starting to sort of go after, looking at, muscles that enable lice to run around, and of course the width and length of the body and a whole bunch of other features. So that's kind of where we're going next with these data. And the goal here, of course, is to really integrate all of these different data sets here to really start understanding, well, what are the selective pressures that have caused these radiations? What are the morphological changes that then have occurred? And how does that manifest itself into those molecular changes across species? Okay, so I kind of mentioned that work that I've done kind of in evolutionary biology and how we kind of integrate tool development into that process. What I'd like to do is talk a little bit about other data mobilization projects that I'm working on. So I mentioned that I'm interested in understanding these broad ecological and evolutionary patterns, both spatial and phylogenetic. Now, with lice, we would never dream to be able to build a map that looks like this. The data is so, in fact, scarce and generally only has country level, and often for many species, only a few individual data points. So this is really a massive lack of data that we're getting in the louse world. So what we've done is apply to NSF for a large digitization project. This is our project called the Terrestrial Parasite Tracker, and our goal is to mobilize 1.2 million parasite specimen records from 28 institutions, and we're about a year into this grant. And the idea here is that many, in fact, millions of specimens are sitting in museums. It's estimated that there are about 1.2 billion specimens, either in the US, 3 to 4 billion specimens worldwide in museums. And the data for these specimens is in fact locked on these little images for lice in particular, on these slides. So you can see from this image, you can see the genus and species of the louse. You can see the host information, and in particular, you can see locality information. So this says granite outcrops, sagebrush, juniper, East Base, Granite Mountain, Tooele County, Utah, at about 4,500 feet, right? And you can even see the date that it was collected in 1966. So what we really want is not just the images of the specimens, right, which is the big goal of this project, but we also want to liberate this kind of locality information so we can start turning that into what are called georeferences, where we get actual latitude and longitude. So this is a project that I've been working on now for a little while, and this is Notes from Nature. I've been working on these projects for a little while. It's kind of surprising when I think about it out loud to a whole bunch of people. So this is a public engagement project. This is called Notes from Nature. It's run through Zooniverse out of Chicago. Zooniverse is the #1 place in the world to go for public engagement projects. Community science projects used to be known as citizen science, but we prefer the term community science now. There you can go and help, identify planets, right, in outer space. You can help people look through camera trap data and identify organisms that they're finding in their backyard, right? Like, this is a really fun website. I encourage everyone to go there because there are so many different ways that you can help and be involved in research. So our project, Notes from Nature, the goal here is that we're working with the public to help us transcribe this museum specimen data. In particular, what we're really interested in is date and location. So essentially what we do is we give a series of images to volunteers, and then we say, all right, help us type out what is the genus and species name, what is the host, right? Who is the host? What is the locality information? This is just called like a locality string, like a string of data, right? That helps us kind of figure out where these things were. So as Larissa kind of mentioned, this project has been going for a while now. We've transcribed over 2 million specimens from over 10,000 volunteers, but to be honest, we haven't actually done a tally in a couple of years, so I suspect that number is quite a bit higher now. They're being transcribed regularly every day. We have some volunteers that are on there every single day transcribing hundreds and hundreds of specimens, and it's been a real rewarding and fun project to be involved in. So we walked through the process of taking an image of specimens. Now we walk through the process of how we liberate the data, but we're not quite there, right? To build a map of specimens. This locality information then needs to be georeferenced. That's where you take this information, somebody goes to a map, hopefully someone that knows Tooele County, Utah in this area, right? And drops a pin right on where they find those granite outcrops, right? And then gets the actual latitude and longitude from that information. So that's called a georeference. It includes often a little bit of error around that, like how much do you trust that data point? So what we're also doing as the next step is developing what's called a gazetteer of localities from georeferenced data. So as you can imagine, this process of someone going to a map, right, dropping a pin down takes a very long time. And when we're dealing with billions of specimens, that's incredibly slow. There are some automated processes that work, but often they need to be double checked, and they work really well for some areas, but are not quite globally ready for prime time yet. Although these processes the development is working really fast. So thousands and thousands and thousands and maybe millions of specimens have in fact been georeferenced already. So the digitization projects, at least in the United States, has been going for about 10 years now. And many collections have already gone through this process of georeferencing the data. So we were kind of thinking like, well, if you have a locality string like this one here, and someone's already gone through the process of georeferencing, well, if another specimen comes in with the same locality string, we don't want them to actually need to go through the process of georeferencing. What would be nice is if they could just put this locality string into a database, what we're calling a gazetteer, right, of localities, and ask, has this record been georeferenced already? And so that's what we've been doing. We just received funding actually to develop this gazetteer, and we've gone through the first initial processes. We've taken all the data from online repositories like GBIF and iDigBio to kind of ask questions about how many specimens have been georeferenced. And if we look at all the specimens that are in these databases that don't have a georeference, but have that locality string, we can say, from the ones that already exist, how many could we automatically populate? How many could we fill in with already previously georeferenced data? And what we're finding is that about 20% of the data of locality strings have been georeferenced, which maybe seems very small, but when you're dealing with billions of specimens, that's 20% ends up turning into a very large number. So that's actually a very exciting kind of project that we're working on now. So our goal here is to just reduce the time and effort it takes for museums to have full georeferences for their specimens. And taking this all the way back to the process of trying to get louse data, our hope is that eventually we'll be able to actually start building out, right, some of these maps of the louse information and start really asking broad scale spatial questions as well as some of these evolutionary questions. All right, so I'll end it there. There's a lot of people to thank in my lab, my graduate students, Avery Grant, Chandra Earl, 3 postdocs, Jessica Oswald, Stanislav Kolenchik, and Bobby Del Carlo, and my developer, Rafe LaFrance, who makes everything work and solves all of our problems, and of course, many, many collaborators, NSF and Universe. So with that, I will end it and I guess take some questions. Thank you.
Great, thank you so much. That was by far one of the most amazing talks I've seen. That was really neat from so many different perspectives, one being just the science and the coevolution, but also the citizen science, the public engagement and all of that, and also just the streamlining of methods and tools and techniques. So thank you so much. So we have several questions that have come in.
Great.
One
Should
is-
I stop sharing? I'm gonna stop sharing, I think, maybe?
Sure, that's fine.
Okay, great, yeah.
One is from Dr. Antonis Rokas. He says, great talk. I have lots of questions, but here are just 2. The first is, how many species of lice are known? And then the second is, does the small genome size of lice mean that they've lost many genes? And if yes, are there particular trends guiding these louse?
This is the best question. So right now there are 5,000 species known, to answer your first question, and it's estimated that that's barely a drop in the bucket. I mean, if you think about the fact that there are 10,000 species of birds known, and many of these birds have different genera of lice on them, like you can imagine the actual reality of the numbers of lice are orders of magnitude larger. Yes, so questions about genome size. So we know that lice themselves have lost their wings and they have really rudimentary eyes. So we know there are some features of these insects that have been lost, and it's very likely those are some of the genes that have helped them streamline their genomes. So lice have the smallest genome, or they had the smallest genome until someone sequenced the stupid Arctic midge, which ended up having an even smaller genome across insects, right? And it is definitely thought of that they've reduced a lot of those features. What's neat though, is the bacteria have done the same thing. So those obligate endosymbiotic bacteria, we've been able to identify multiple independent obligate endosymbiotic bacteria, and they have all lost the same features over time to reduce those genomes. So I think we're seeing that pattern in lice, and maybe that just happened once at the origin of lice, but when you look at their endosymbionts, you see that pattern happening over and over and over, which is weird and cool.
Wonderful, great, thank you so much. So we have another question from Jacob Stenwick, and he says, I can imagine ATRAM will work well for single copy genes. How does ATRAM perform on assembling multi-copy genes? And will this be a problem for fishing out the 13,000 query genes in the new reference louse?
Yeah, it's a really good question. In particular, this is a big question for plant people who think about like who have like megaploidy, right, with plant genomes that have been duplicated millions of times and trying to figure out what copy is what. So you're absolutely correct. ATRAM certainly works the absolute best on single copy genes, and because lice have the smallest genomes, we're mostly just dealing with single copy genes. So it works well. We have used ATRAM on a whole bunch of different organisms with great success. We have, in fact, over the last year, started working on a few pipelines for dealing with paralogy. In particular, the idea is sort of like seeing how many contigs are assembled in ATRAM and then building trees with those to sort of look at, we think this contig then is likely this paralog and this contig is that paralog. So trying to really start teasing that apart and fishing that out. I mean, it's a huge, huge question for phylogenetics for many copy genes. Thanks.
So I have a question, and I couldn't get the image of like T-Rex with feathers and lice out of my head while you were talking about it. and on that note,
I put that in there for you, by the way, Larissa.
Wonderful, thank you. Okay. So I was just trying to, do you think that this could potentially help us understand relationships between things that we can't get ancient DNA of, such as some of these ancient dinosaurs, or what are the kind of prospects there in exciting directions?
Yeah, I mean, one thing that's really neat, when you look at that tree of lice, you see that there are many lineages that actually went way back before the extinction of the dinosaurs. So it might give us an idea of, how many ancient dinosaur lineages had feathers at that point, right? Like that maybe we didn't know about before. Certainly we only know about the ones that we find, right? And then we can kind of extrapolate onto like birds now and where they might have come from. But the actual diversity of lineages that have been around for a lot longer really does lead us to some really interesting questions about how many feathered dinosaurs were there? What were their relationships with their lice, right? Like, do they have bills? What does that look like? Like there's a lot of fun things to think about.
Very cool. Well, on that note, and maybe the suggestion to Luis Ray to include some lice on T-Rex, who's a scientific illustrator, we will continue with any Q&A during our virtual reception. So our virtual reception, the link was sent out in the chat. So please join us today. That's a chance for all of us to be able to see each other and talk one-on-one. So that will start at 4:15 Central Time today in just about 14 minutes. So thank you so much for this talk. It was really amazing, and I look forward to talking more in the virtual reception.
Thank you. Thanks a lot. Thanks for inviting me. Thanks for coming, everyone.
Okay.